Hello everyone, welcome once again to this presentation on protein. We are now in part 3 of this series of lecture. So today, I'm going to discuss with you the 4 types of protein structure. In some other authors or in some other textbook, they call it 4 levels of protein architecture. Okay? Or 4 levels of protein design. So here, the 4 types of protein structures are the following. The primary structure of protein. So what is primary structure of protein? So it says here, it refers to the um, order in which amino acids are linked together in protein. Remember in the previous slide, I showed you how peptides are formed, okay? Or how amino acids are linked together in a peptide. So peptide, we use the term when we are referring to short chain of amino acids. If it is oligopeptide that refers to the chain that has, say, less than 10 amino acids. Okay, but when uh, but when there are more than, okay, more than if there are uh, hundreds of amino acids linked together, or even less than 100 of amino acids together, you call that polypeptide, okay, or also known as protein. So these amino acids in proteins are linked by peptide bond, okay, or amide bond. And remember that when we read the sequence, when we identify the sequence, we begin from the N terminal to the C terminal. I explained to you already that the N terminal amino acid residue is the amino acid residue that has free amino group, whereas the amino acid residue at the C terminal is the amino acid that has free carboxyl group. So the N terminal is at your left side, okay? If you identify from left to right, the N is the amino acid at the leftmost, and then the C terminal amino acid is the amino acid at the rightmost. Okay, so proteins uh, are made up of amino acids, and each uh, each protein has their own sequence of amino acid. Like for example, this insulin, okay, I have shown you this in the past. Okay, this insulin is made up of 2 chains. Okay, so there are 2 polypeptides here, polypeptide A and polypeptide B. The polypeptide, the 2 polypeptides are linked together by what we call disulfide bridge or disulfide linkage between the cysteine in chain A and cysteine in the chain B. And there's also another disulfide bond on this region. Okay, so this is the primary structure of insulin. Now look at this table. Why is sequence important? There are 3 species here: human, porcine, and bovine. Okay, and these are the sequence. This is the sequence of of the amino acids. Okay, in chain A, in chain A, the the eighth amino acid in human is threonine. Okay, where is that? Where is that eighth amino acid? Okay, in if we count if we count the sequence beginning from glycine, okay, the eighth amino acid is alanine. So this is the eighth amino acid. So we suppose that this sequence is or this insulin is the sequence of bovine or cow, because the 8th amino acid is alanine. However, in pig and in human, this alanine is replaced by what? By threonine. Okay? So the amino acids before the 8 amino acids, human, pig, and cow share the same sequence. Okay? Now, of course, my insulin and your insulin, we expect that we have the same sequence of amino acid. In in the ninth amino acid is serine. So we don't differ, okay, in the amino acid residue. The 10th amino acid, okay, the 10th amino acid here, this is the 10th amino acid. It's valine, okay. In human, this is replaced with isoleucine. So again, this is the primary structure of um, bovine albumin. Let's go to the chain B. The 30th amino acid um, in human is threonine, but in pig and cow, the 30th amino acid is alanine. Where is that 30th amino acid? This would be your amino acid number 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30. The 30th amino acid in cow is alanine, same in pig, and it's replaced by threonine in human. So what I'm saying here is, uh, although the same protein, okay, insulin, but they differ in the sequence in different species, okay. However, in some cases, although, for example, in human, same protein, but the amino acids are altered, and it causes serious illnesses, okay. The next level of protein structure, or next level or second level of protein structure, is what we call the secondary structure. It says here that this structure arises from hydrogen bonding between atoms of the polypeptide backbone. So this structure exists or happens because of the hydrogen bonds that are formed between atoms in the polypeptide backbone. Now, first of all, what is hydrogen bond? Hydrogen bond is an attraction between, okay, electronegative atoms such as fluorine, oxygen, and nitrogen. The hydrogen bonding is not a covalent. It's not a covalent bond. Remember, covalent bond is attraction, okay, that is due to sharing of electron. Hydrogen bonding is a force, or it's an attraction that takes place between this is partially positive atom, okay, hydrogen is partially positive atom, and these atoms here are partially negative. So that's why there's an electronic attraction between the partially positive hydrogen and these atoms here, which are partially negative. Okay, we know that these atoms are electronegative atoms. So Again, these structures, the secondary, arises from the hydrogen bonds that are formed between the atoms in the polypeptide backbone. So, what are the atoms in the polypeptide? Okay, in the polypeptide backbone, or what are the atoms in the backbone of polypeptide? You would consider these atoms as backbone. Okay, backbone is this supports an organism. Um, in protein, this backbone also supports the structure of protein. So what are the atoms in the backbone? Okay, you would consider this as a backbone, the C, CO, okay, and then the N here, all right, then another C, another CO, okay. Those are the atoms of the backbone of the polypeptide. So when when hydrogen bonds take place between those atoms, this structure, these structures arise. There are two types of secondary structure, the alpha helix, which is a structure that is adapted by a protein, okay, that assumes helical shape. It says here, a single protein chain adopts a shape that resembles a coiled spring. So this is the structure of alpha alpha helix. So take note that there's a hydrogen bond between atoms in the polypeptide backbone. So where are those? Where are those hydrogen bonds? Take note that this is the carbonyl group. This is the CO. Okay. These atoms here, this CO, this is part of the um, backbone, atoms in the backbone. And then this oxygen here, remember as I said, oxygen is electronegative, and this oxygen here is attracted to the hydrogen in the amino group. Okay, this is the amino group. Remember this amino group here, this nitrogen here is part of the backbone. So when this hydrogen, which is partially positive, is attracted to this oxygen here, okay, um, what result is a helix. This region here, or the hydrogen bond, I should say, the hydrogen bond between atoms happens every um, every fourth amino acid. So for example, if this is the first amino acid here, okay, if there is an amino acid here, this amino group here would be the amino group of the fourth amino acid. Okay? So the hydrogen bond happens between the carbonyl group of the first and the amino group of the uh, you have to count uh, in between hydrogen bond there must be like two amino acids. So for example, this carbonyl group here, right here, this is the carbonyl group of the, say, second amino acid. This carbonyl group here will form hydrogen bond with the amino group of the, this time, is not the fourth amino acid but fifth amino acid. Okay? If this right here, if this carbonyl group here is part of the third amino acid, then as you would see, this is attracted to this amino acid, or amino group of this amino acid, and we would assume that this amino acid is the seventh, okay, seventh amino acid. So that's what alpha helix does. Okay, the second one is beta pleated sheet. it says here it assumes a flat, no, flat or completely extended amino acid chain. Okay, it resembles a sheet. Now, the hydrogen bonding between two different chains happens. It could be inter or intramolecular. Intermolecular, okay, intermolecular in between these chains, okay, or when we say intra, within the chain. I want you to take note that in secondary structure, the side chains are, or the side chain do not participate in the formation or in the structure. In the primary structure, the side chains are outside, okay, outside the coil. There is no enough room, there's no enough room for the R groups. So in order for them to fit inside the coil. That's why they are outside the helix. In the beta pleated sheet, the R groups are either below or above the axis. Okay? So they do not, in other words, the R groups in secondary structure do not participate in the formation of the structure. Okay? It's only the hydrogen bonds between atoms in the backbone that maintain the structure. Next is what we call the tertiary structure. Okay? Um, Tertiary structure is a result of overall folding, overall three-dimensional shape of a protein. I want to repeat again, reiterate again that in the secondary, the R groups, okay, or the side chain do not participate in the maintenance of the secondary structure. It's only the hydrogen bonds of the atoms in the polypeptide backbone. But in the tertiary structure, this results from the interaction of the amino acid side chain. Okay? What happens here is this structure, this structure happens, the tertiary structure happens due to the interaction of the side chain of the amino acids. Okay, these amino acids are widely separated from each other, but um, they exhibit forces. Okay, now what are the, so we can say that in order to maintain, in order to stabilize this tertiary structure, it's actually the side chain or the R groups that govern or maintain this structure. What are the interaction? What are the attractive forces that maintain this structure? As you could notice here, the interactions could be salt bridge, hydrophobic interaction, disulfide, hydrogen bonds. So these are the four attractive forces that maintain the tertiary structure. So how do this how do these attractive forces happen? Okay, that happens when amino acids are what? Amino acids are attracted to one another depending on what is their R group. Remember in the past that we have learned the four or the classification of amino acids based on the side chain. Okay, what are the, if you would recall, what are the um, classification of amino acids based on R group. We have non, we have non-polar, okay? We have polar, we have polar neutral, okay? We have polar acidic, and then we have also polar basic. Non-polar, due to the presence of what? Hydrocarbons in their side chain. Polar neutral, the reason why they are polar because they have atoms that are electronegative. Okay, recall what are the amino acids that belong to these categories. Polar acidic because the side chain is acidic. Polar basic, the side chains are basic. So it's your again responsibility to go back to those classification. So when those amino acids that belong to these categories are attracted, what results is this attractive forces. Okay, let's consider those attractive forces one by one. So disulfide bond. What is a disulfide bond? Disulfide bond is the strongest among the four because this is covalent. What is covalent? Sharing of electron between what? The two cysteine groups. Okay? Look at this. In this region there is cysteine. Okay, there is cysteine residue, and then in this region also of the protein there's also cysteine. Okay, remember the structure of um, the side chain of cysteine. Okay, this is the R group. Let's put the N here, the amino group. Okay, we can do this this way. Okay, this is the structure of amino acid cysteine. Okay, we call that cysteine. However, when two cysteine, when two cysteine residues form a disulfide bridge, we call these amino acid residues cysteine. Okay, cysteine. Cysteine, okay, cysteine, this is this refers to the individual amino acid, the reduced form. Okay, this is the reduced form of the cysteine amino acid. However, when two cysteine residues combine, we now call them cysteine. Okay? So the disulfide bridge happens again between two cysteine residue. So this is the disulfide linkage, disulfide bridge. You notice this is a solid line. That means this is covalent. All right. Next is electrostatic attraction, electrostatic interaction, or salt bridge. This happens when there is, or um, the neighboring amino acids are what? Or have um, acidic side chain or basic side chain. Okay, I repeat, electrostatic interaction happens if or where there is or there are amino acids that are acidic or and basic amino acid. What are the examples of acidic amino acids? Okay, let's say this is aspartate or aspartic acid. In this region, there is aspartic acid. Okay, on this region, there is what? Let's assume that this is lysine. Okay, lysine. Remember, lysine is basic and aspartic acid is acidic. So when they are close to one another, they form what we call electrostatic attraction. Being basic and this is acidic, so they form what we call salt bridge. Okay? So all you have to remember is this electrostatic attraction happens between amino acid that has acidic side chains. Anyway, there are only two amino acids that have acidic side chain, um, glutamic acid and aspartic acid. So those two can form electrostatic attraction between lysine, okay, histidine. The third one, the third type of interaction that govern the tertiary structure is called hydrogen bonding. Okay, hydrogen bonding, as I said earlier, this happens between the hydrogen atom and the this is attracted to either fluorine, oxygen, or nitrogen. So for hydrogen bonding to occur, the hydrogen must be attached. Actually, must be attracted. Let's say must be attracted, not necessarily attached. Okay, must be attracted to the oxygen, nitrogen, or fluorine. So, what are the amino acids that can form hydrogen bonding? Those amino acids that are polar, okay, that are polar, those that have oxygen, nitrogen in their side chain, okay, can form hydrogen bonds with the other polar amino acids, okay? And the last one is hydrophobic interaction, hydrophobic between non-polar side chain. And we know that there are several amino acids that have non-polar side chain. For example, this, okay, attraction between two amino acids that are attraction between two valine. Okay, this is valine, and this is also valine here. Okay, valine, val. This is also a val. Okay, their side chains are both hydrophobic, so they are attracted to one another. Usually, the hydrophobic interactions, if the protein is soluble, okay, let's say this is the protein. As I said earlier, the hydrophobic interactions, okay, are abundant in the core of the protein. Because on the surface, what you see there are the polar amino acids, but the non-polar amino acids, they are usually in the core of the protein, okay, because they hate water, they don't interact with water. Okay, the fourth level, which is the highest, is called quaternary structure. Okay, in this structure, the assumption is a quaternary structure of protein refers to the organization among the various peptide chain in a multimeric protein. Now, I'd like to focus first on this protein, okay, in this picture, so that we can illustrate what is quaternary. The whole thing, the whole thing is a protein. In fact, this is the protein of hemoglobin. Hemoglobin, as we know, carries or transports oxygen to the different parts of the body. There are four polypeptide chain. You call that subunits, okay? Um, Brown, orange, pink, and then purple. So there are four polypeptide chain. Okay, each color-coded subunit is a polypeptide. Okay, however, these four polypeptides, okay, are attracted to one another, and they are considered as one protein only. Okay, so we call this protein multimeric. Why multimeric? Because there are four uh, polypeptide chain. Okay, so this is the highest level of protein organization. So in order to classify in order for a protein to be classified as quaternary, there must be two or more polypeptide chain. There must be two or more subunits. If there's only one subunit, then that is not a quaternary structure. Okay? So not all proteins have quaternary structure. Okay? But all proteins, of course, they have what? They have primary structure because they have sequence of amino acid. Okay? The subunits the subunits are generally independent of each other. Okay, independent. So we can say that these subunits here are independent with one another, but they are not covalently bonded. So this purple here, is not covalently bonded to this polypeptide, but they are attracted. Okay. Proteins with quaternary structure are often referred to as oligomeric protein. Okay, oligomeric protein or multimeric protein. Okay, they contain even number of subunits. Hemoglobin, as I said, there are four polypeptide chain, and for your information, this tiny material here is the prosthetic group. Okay, remember that proteins are also, I should say, classified based on the components. Could be simple or conjugated. Okay, this is a conjugated protein because there are non-amino acid component. This region here is the heme group. Okay, heme. Okay, let me just write here heme. In the heme group, that's where the oxygen binds. Okay, so these are the four levels of protein structure: the primary, secondary, and tertiary and quaternary.